scale at which Ben is able to measure everything, so I'm going to try and do my best to justify how we can still learn things from a smaller number of mutants. Okay, so starting at the basics, there are a variety of physical forces that come together to dictate for a given sequence how it folds into either a folded protein or some given conformational ensemble. And we can describe that with a thermodynamic constant. And then these protein ensembles go on to do jobs throughout the cell, perhaps binding DNA or other proteins or enzymes that catalyze a particular chemical reaction. And again, we have kinetic and thermodynamic constants for this. And then the entire sea of all of these proteins working together are what determines the state of an individual cell. And so I think it's a dream of probably many people in this room to eventually be able to have some kind of algorithm that would allow us to start with the sequence of a protein and not just predict its folded structure, but predict all of these different functions that it's going to carry out. And it seems like a pretty hopelessly complex problem, but I think that we can draw an analogy and some inspiration from other fields where they've really seen tremendous success. And so there's this book called The Weather Machine, and it talks about the history of weather prediction. And it starts with this guy, Lewis Fry Richardson, who people before him had kind of realized that weather was just fluid mechanics in the sky. And he realized that maybe we could take this system of coupled differential equations that was impossible to solve, and we could break it up into tiny little bits, where there would be like a giant concert hall with 300,000 people and a single conductor who would assign each person a different area that they would do the calculation. And with this system, it would take 24 hours to calculate the weather 24 hours in the future. So he has this quote where he says a little over 100 years ago, perhaps someday in the dim future, it will be possible to advance the computations faster than the weather advances and at a cost less than the saving to mankind due to the information gained, but this is a dream. So you fast forward, and we started to have these computers that were incredibly powerful. Now you have computers more powerful than this in your pocket. And now, with the development of all of these climate models, this was an article just from last week where not only is the weather really accurate 72 hours in the future, far beyond what he was imagining, but we're actually approaching a place where we could be looking weeks in the future with some accuracy. And so when we think about what drove this incredible progress, the first was advances in computational power, but we have that too for our problem. The second was advances in these climate models, but we also have that from many of the people in the room right now. And then the third, I think, was that there was this network across the world where each of these red spots is a different weather station, where they were measuring the weather, and they were doing it on an absolute scale. So instead of talking about the fact that the weather, it's 2 times hotter than it was yesterday, or it rained 10% more this month than last month, they were using this absolute language of physical constants that made it really easy for them to take all of the data across space and time and aggregate it to train the models. And so I'm arguing that I think that we should move towards a similar thing, thinking about proteins, in terms of being able to actually measure some of the impacts of mutations on function using absolute kinetic and thermodynamic constants. And so how can we do this? If we think about the way that we've traditionally done some of this biochemistry and biophysics, there's just no way that we're going to be able to scale these giant flasks to measure the number of proteins that we're going to have to measure. And so for the last decade, my lab has really focused on developing a series of microfluidic platforms that could allow us to recombinantly express, purify, and characterize many different proteins in parallel. And so we have 2 platforms that I'm not going to talk about today, some spectrally encoded beads that allow us to do some small library-on-library measurements, as well as these microfluidic double emulsions for ultra-high throughput sorting. But today, I'm going to tell you a story that's just using these valved devices for characterizing enzymes. And so this story is a story that's really being led so far by Albert Lee and Daniel Mukhtari, both of whom are really talented people who have worked in my lab. And it's a close collaboration with Dan Hirschlag's lab at Stanford. And it's all unpublished, so we'll see how it goes. Okay, so we've been talking a lot about whether we could classify a given variant as being benign or pathogenic, but really we'd like to know more. we'd like to know, how does a given variant impact different aspects of molecular function? Do variants with similar biochemical signatures end up driving really similar disease phenotypes? And then finally, could we generate therapies that would allow us to restore specific biochemical signatures that have been altered by mutation? And so today, the story I'm going to tell you talks about this protein, SHP2, which is a protein tyrosine phosphatase. And SHP2 is a critical signaling protein that lies upstream of the RAS signaling pathway. And it's also part of this PDL1, PD1 signaling pathway, where it's downstream of that. So it's important for thinking about developmental disorders and cancer. And mutations in this protein have profound effects. So these cause the majority of these developmental disorders, Noonan syndrome and Noonan syndrome with multiple lentigines. They're associated with a variety of juvenile cancers, and they're also associated with metabolic disorders, glucose metabolism. And right now, there are a series of allosteric inhibitors that are targeting SHP2. So this is an attractive drug target that people are thinking about for cancer therapies. If we think about SHP2 at the molecular level, it has kind of this, this protein tyrosine phosphatase catalytic domain here that I'm showing you in tan. And then it has two SH2 autoinhibitory domains. And it's thought to exist in these two states. So either these SH2 domains come over and they kind of occlude the active site so that the protein's in an autoinhibited closed conformation, or they swing open to expose the active site. So we can kind of take that picture from the prior page and we can show it as a bit of simplified model where we've got a protein that's either closed or it's open, and there's this transition between. We know that this open conformation can be stabilized by binding to an activating peptide that prevents it from closing in on itself. So that's one thing. And then we also know from a series of crystal structures for some of these allosteric inhibitors, if you look at these crystal structures, hopefully you can recognize that this looks like the closed state. So these allosteric inhibitors are sandwiching the autoinhibitory domains on the catalytic domain. So now we have kind of this two-state model where we can also think about peptide binding, we can think about drug inhibition, and any mutation in this protein could impact any one of these different transitions. Right. They could also impact thermodynamic stability. I'm not going to talk about that today, but we've measured that too. So a first question is, how do SHP2 allelic variants impact biochemical function? And so there are, as of a couple months ago, there are a little over 400 annotated human alleles. Most of them are of uncertain significance, like most of the talks that we've listened to. And so Albert and Daniel went ahead and they generated a library of 192 single substitution mutants throughout SHP2, so in the catalytic domain and in these autoinhibitory domains. And then they wanted to ask the question of, do disease-associated variants impact catalysis? So they're going to do this using these valved microfluidic devices that we have, where this is a picture of the device sitting on a normal 1-inch glass slide. And each of these devices has 1,800 1 nanoliter reaction chambers, and the fluid flow within and between the chambers is controlled by almost 6,000 on-chip valves. And so the way each experiment works is we start by making a library of plasmids, each of which encodes a known variant. And then we use a robot to print those plasmids into an array. So here I'm showing you 3 different spotted plasmids. There's 1,800 each time we do an experiment. And we know who's who by its position within this 2D array. Then we can align by eye microfluidic devices on top. So now each of these 1 nanoliter chambers is programmed with a different variant. We can push cell-free protein synthesis mixture into all of the chambers on the device and close valves so that the chambers are isolated from one another. And now we just put the whole device on a hot plate and we incubate and we produce protein in each chamber. The protein has a C-terminal EGFP tag, so that's why I'm coloring all of these chambers green. We can then kind of specifically pattern a specific portion of each of these chambers with an anti-EGFP antibody, which pulls down the expressed EGFP tagged protein. And then we can wash We can use the intensity of the pulled-down protein to calibrate exactly how much protein we have in each chamber. So we have like a known enzyme concentration. And then we can iteratively introduce different reagents. So in this case, we're going to introduce this fluorogenic substrate that produces light when the enzyme cleaves off a phosphate, when the phosphatase does its job. And we're going to monitor the accumulation of light in each chamber over time. And so all of this, these are what the devices look like when they have all the tubing plugged into them. And all of this just sits on top of a commercial automated epifluorescence microscope. And this is all just a bunch of custom-built pneumatics where all it does is determine whether we pressurize the valves or not. So we have a computer that can control all of the fluid flow on the device. And the raw data looks something like this. So you can see there's like a series of chambers across the device, and they're all accumulating light at different time. So we can just image, timestamped images, and we can follow the reaction progress. And each time we run an experiment, we get a little image like this that shows what the immobilized protein looked like. We can monitor the amount of protein that remains over time. We have an individual standard curve for every single chamber, so we can say how much product was made, corresponding to how much light we see in that chamber. And then we're able to introduce substrate, where each time we introduce a different substrate concentration, we can follow the accumulation of fluorescence over time, so we can fit this initial rate. We get a rate as a function of substrate concentration, and that's really what we need in order to be able to fit a Michaelis-Menten curve. And there's negative controls in chambers scattered throughout the device. We also go back and we'll measure the same substrate multiple times to make sure that we're still getting the same answer. So there's a bunch of quality control built into the experiments. Okay, so this is kind of what we see in one chamber when we run a device, and each time we run a device, we get about 1,800 of those reports each time we do an experiment. Okay, so coming back to our original question, how do SHP2 allelic variants impact biochemical function? Albert and Daniel went ahead and they measured all of these allelic variants to look at their kcat over km for this fluorogenic substrate. So I'm going to be showing a lot of these volcano plots. We're on the x-axis here, this dashed line is the wild type value. You can see whether a mutant went up or down. And then on the y-axis, we have a bootstrapped probability estimate of the statistical significance with which we can say a variant is different than the wild type. And so you can see here that there are some mutations that make the kcat over km go down, but most of them actually increase the kcat over km. We can plot these on the structure, and we can see that this makes sense. So the things that make the kcat over km go down, they're all kind of in the catalytic domain. They're reducing catalysis. And then the ones that are making it go faster, they're in the autoinhibitory domain. So they're relieving autoinhibition and making it go faster. So then we can ask, do variants impact catalysis, autoinhibition, or both? So Albert and Daniel went ahead and they just made another version where they got rid of the autoinhibitory domain, and they profiled all of the impacts of the mutations in that domain. And now you can see when we look at the volcano plot, most of the mutations reduce catalysis. That's what we would have expected. And now we can compare the impact of a given mutation on the kcat over km in the full-length protein and in the catalytic domain alone. So here are all of the wild-type-like variants that look the same in both constructs. We can see here, these are faster in the full length, so these are all relieving autoinhibition. A lot of these are just kind of on this diagonal line, so they're reducing the intrinsic catalysis. And then we can also see here, there are variants that simultaneously reduce catalysis and autoinhibition. So some of them are going to look wild-type-like in the full length, but you can only kind of uncover these effects when you compare the two. This also allows us to define kind of this fraction open, so we can look and see how a given mutation is impacting the amount of time that the enzyme is in the open conformation. All right, and again, we see sort of what we expect: reduced catalysis in the catalytic domain, reduced autoinhibition in the interface and in these SH2 domains. But when we try and ask, does this really help us explain all of the known pathogenic mutations? It doesn't quite yet. So here are all the known benign variants. Here are the known pathogenic variants, and most of them are relieving autoinhibition. But there's a subset here that are known pathogenic, and they don't appear to have anything to do with autoinhibition. And figuring out what's going wrong here is important for being able to ultimately predict what's going on with a bunch of these other variants of uncertain significance. So now we can start to ask, what about the other functions? So how do these variants impact binding to the activating peptide that holds the protein in the open conformation? So here's the sequence for one of these known doubly phosphorylated activating peptides that binds to both of the SH2 domains. And Albert was able to go ahead and remeasure this rate, where now he's looking at the rate at a single substrate concentration, and he's systematically varying the amount of activating peptide. And now he can compare, again, we've been doing this all along, where we compare for a few variants the measurements on the device to what we see in a plate reader. And for this activation assay, now he's getting similar EC50s. And we can look, okay, what about all of these human allelic variants? How do they impact binding to the activating peptide? And you can see here, most of them are actually tightening the affinity to this activating peptide. And that makes sense when we think about this model, right? Because any mutation that makes the overall complex more open is increasing the effective concentration that's available to bind the activating peptide. So we would expect to see this relationship. And deviations from this relationship should tell us any mutations that have an altered affinity to the peptide. So here's what the model predicts. If we compare the EC50 to the fraction open, we should see this linear relationship, inverse. And that's mostly what we see. But there are some outliers here. And what's kind of gratifying is that many of these outliers were some of these same variants that before we weren't able to explain just from looking at autoinhibition alone. So now we've explained what's going on with these, and we've also provided additional information about other variants that are likely to have a similar pathogenic effect. Okay, so can these biochemical signatures more accurately predict impacts of variants of uncertain significance? So now we can kind of take, here's the known clinical annotation, but now instead of just saying whether something's benign or pathogenic, we have kind of a readout of this biochemical signature that we can cluster to group mutants according to how they behave. We can compare this to alpha missense, and here, similar to some of the other discussions, there's all of these mutants here appear to have a pretty similar biochemical signature and cluster with the benign variants. Alpha missense seems to overpredict some of these as pathogenic, although maybe they have localization defects or other things that we wouldn't be able to probe in our assay. And we can use this information to try and come up with a logistic regression that allows us to predict the likely impacts of additional mutants. So we can do that, but we can do some other things that I think are particularly interesting. We could ask, what about drug response? Do any of them have an impact on the sensitivity to drug? So here, we can look, again, kind of like the activating peptide. We can now look at the measured rate as a function of increasing amounts of an inhibitor, and we can fit this to a curve. And we can see for different inhibitors on chip, we can see a similar inhibition curve and get out an IC50. And these are largely in line with what's been seen before in literature. But what becomes really interesting is now we can look at how do these inhibition curves differ for all of the different human allelic variants. And now you can see we're still collecting additional replicates here, so we don't have the significance that we need to have in order to publish. But we're already beginning to see that several of these variants are actually increasing the IC50 by 2 orders of magnitude. And so now if you were trying to classify patients for a clinical trial, these are patients that you would want to eliminate, right? You wouldn't want to have them. And you can see, some of these mutations here are known cancer hotspot mutations. So these are actually relevant for thinking about patient care. So pretty much everything that we've talked about before, I think could have probably been done in a cell-based assay. I don't know that you necessarily needed to have the in vitro measurements. But here we've taken the time to run, we only have 192 mutants, but we have about 350,000 different reaction progress curves. So what does this uniquely allow us to do? One thing we can do is we can go back to this model of SHP2 function activation and inhibition where there are two states, closed and open, and the drug binds to this closed state. In this model, it makes a few quantitative predictions. So first, it says that at high enough drug concentration, there should be zero residual activity, right? Because everything should be pushed to the closed state, and the closed state has no catalytic activity. The second thing is that any mutation that decreases the fraction closed, that holds it more open, should also make the drug bind more weakly, right? And so we can test this first prediction that there should be no activity at very high concentrations. I kind of showed you these IC50 inhibition curves, and I did what is really common, where I just normalized the top to 1, and I normalized the high drug concentration to 0. If you don't do a double normalization, you see it makes sense to normalize to 1 because these different variants all have different intrinsic activities, and we want to be able to compare them to one another. But for all of these variants, if we don't normalize to 0 the very high drug concentration, you can see that in most cases, there's still really appreciable residual activity, about 50% of the activity, even at the highest drug concentrations. We can also test this idea that if the proteins are more open, then we should see a weaker IC50, right? So as the fraction closed goes down, the IC50 should concomitantly drop. And when we put all of the data comparing these two parameters, we don't see that relationship at all. So now we have two pieces of information that tell us that something's wrong with our model. And these two pieces of information imply that there must be a state that isn't fully closed, that's able to bind the drug, and that still has some activity. We can go back, and the model also makes some predictions on the other side when we're just thinking about activation, where I was telling you we should have kind of this inverse relationship where there's some variation in peptide affinity, but largely the EC50 should vary inversely with the fraction open. And I showed you this plot before, and one thing that I didn't really highlight was this isn't totally true. So when we look at these enzymes that are more closed, the EC50 is really systematically higher than it should be for these very closed enzymes. And this means that some activity must come from a state that isn't fully open and that can't bind peptide. So when we take all of this information we have from this quantitative modeling, we can take this initial model we had of kind of this two-state system, and now we know we have to add an intermediate state. And I realize this is getting a little bit complicated, so hang on a little bit. But we know we have to have an intermediate state that can bind drug, and that can an intermediate state that can bind drug, and it can also bind this activating peptide. And there are some other orthogonal pieces of data where people have done single molecule FRET looking at the motion of these SH2 domains that have suggested that there might be this intermediate state that isn't open and it's not closed. And we can test if this intermediate state exists because this three-state model makes a few different predictions. So the first prediction it makes is as we add activating peptide, we should be able to shift the population to different states. So we have an activating peptide that preferentially binds the N-terminal SH2 domain. It still binds, but it has a weaker affinity to the C-terminal SH2 domain. So if we start with variants that are mostly closed, as we increase the amount of this activating peptide, we should pull the cycle here where we see first more intermediate states, and then finally we get everything into the open state, and we can begin to see the binding of the peptide to this second SH2 domain. And so now if we know that, if we can pull the entire equilibrium to the intermediate and open state, we should see that initially when we add drug, in the presence of this peptide, the IC50 goes down as we have more proteins that are in this intermediate state, and then it starts to get worse again. So binding the drug should get tighter, and then it should get weaker. And we can go ahead and we can simulate what we expect. So if the two-state model is true, we should just see an increase in the IC50 as a function of this activating peptide. But if the three-state model is true, we should see an initial dip and then an increase, and this increase represents binding to the second SH2 domain. And when Albert goes ahead and makes these measurements, that's exactly what he sees. He sees this initial dip, and then he sees this increase. Okay, well, now we can make another prediction, where if we get rid of this binding site, we shouldn't see that final increase, right? It should just go down and it should stay there. And luckily, there are some prior publications that have characterized this mutant, R138A, that has a defect in the binding site in that C-terminal SH2 domain. And so now when we do the simulations, what we should see is this initial drop in the IC50, and then it should stay constant because we no longer have that C-terminal binding. And that's exactly what we see for this R138A data. And finally, what we can do is if there are mutations that already favor the intermediate state, we shouldn't see that initial dip because we weren't initially in the closed state, and so we're not going to be able to pull it that way. So we should only see an increase in the IC50. And so luckily, there's a mutant here, D61G, which we had already shown was preferentially in this intermediate state. Now when we do the simulation, we expect to just see an increase in IC50 as we add this activating peptide, and that's exactly what we see when we do the experiments. So who cares? Like, why does any of this matter? And I think the reason why this matters is there are real consequences for having a catalytically active intermediate. So any drug that binds this intermediate is going to have limited efficacy because it doesn't matter whether you're a picomolar binder or a nanomolar binder, there's still going to be a large population of molecules that are still turning over, and you won't see a loss of activity. And so that means that all of these drugs that are currently in development are unlikely to actually completely inhibit the protein. So I hope that this has maybe convinced you that high-throughput biochemical data can provide rich information about which functions are disrupted by mutations. We can use this to identify potential non-responders in clinical trials. We're hoping that these types of data can be used in training a next generation of models to predict protein function from sequence. And hopefully some of these functional data can reveal the existence of states that are really hard to get at using structural techniques. And I just wanted to highlight really quickly, there's one student from my lab here, Micah Olivas, so he gave a poster yesterday. If you didn't see it, I encourage you to find him and talk to him about his work. And I just want to thank my lab and funders and you for your attention.